So unlike the past speakers, Viviana Gradinaru is new to the Institute. So I actually met her for the first time last night. So, but it's great that she's here as we're thinking about the future in the next decade. As I mentioned in my prologue, the Allen Institute is very excited, as is the field in general, about the new tools that are becoming available. And Viviana will be telling us about using some of those bleeding edge tools in her research program at Caltech. So welcome, Viviana.
Thank you. Can everybody hear me okay? And let's see if this projects well. Great. So thank you, Dr. Gibbs, for the introduction. I will stay true to my career stage and show lots of data. So let's start with a graph. It's a population pyramid, and what this does is to represent males on the left and females on the right by age and numbers. And these population pyramids have been looking more and more interesting recently across the globe, and if we look in time as well. So this is from a well-developed nation. I would say it's not United States, but it's from a very well-developed nation. And if we look at the numbers for 2007, what you can see is actually very interesting. It's not a pyramid anymore. And if we project, if we fast forward into the future in the spirit of the symposium, the expectation for 2050 is rather interesting. It is a reversed pyramid. And the numbers are very telling, and this is what we see with some of the developed nations nowadays. And this is somewhat worrying, right? Because we know, at least as neuroscientists, that these emeritus brains here are very valuable to society. You don't want to only live longer, you want to live well as well and contribute. And the unfortunate truth is that many of these brains are affected by Alzheimer and Parkinson. So it's a very, very serious problem, and it points to the fact that we need to understand the brain, and also that we need to understand the aging brain with the increasing lifespan. And just to give you more numbers, it's projected that from 2005 to 2030, there's going to be a doubling in the number of Parkinson cases. So this is all very worrying and points to the fact that we need to understand the brain, but the reality is that sometimes we don't have the appropriate tools. So we need to invest in building these tools with the hope that they can at first be used for basic science to understand the brain, but then they could help for translation purposes as well. One such example where a research tool, and I think David Anderson very nicely illustrated in the opening talk, electrical stimulation has been used since early 1900s in animal models of disease to understand brain circuits. And here what you see is the Canadian neurosurgeon Dr. Penfield, who used electrical stimulation in his epileptic patients and mapped the cortex and made correlations between stimulated different brain areas and motor actions or sensations. And not only for basic research purposes, this technique actually found its room in clinics, and it's very successfully being used for deep brain stimulation on Parkinson patients. So electrodes are implanted into the deep brain structures, and you zap the motor structures at very high frequency, and this restores motor symptoms very successfully. However, there are side effects that are not that unexpected, given that the brain is highly heterogeneous. And as David was mentioning, electrical stimulation is very likely, because of the properties and the temporal parameters of the stimulation, to activate axons of passage or axons onto the structures. So the stimulated structure doesn't even become that relevant. You need to understand the brain in its old context with the connectivity. And this is why the work at the Allen Brain Institute has been so revolutionary, because it pointed us to the fact that there is a lot of content into a nucleus beside the cell bodies in that area. So to understand, to tackle some of this complexity, new tools were needed. And as it was mentioned earlier, optogenetics is such a tool. And of course, methods for chemical control and temperature-based control as well. But I will focus on the optogenetic methods in which I was involved developing. So what you see is electrical stimulation here could affect all cell types in the vicinity of the electrical field, so there's no specificity. And this is cause for the side effects with electrical stimulation. Some Parkinsonian patients that have deep brain stimulation report disturbing side effects such as a tendency to go to Vegas and gamble all their money, or inappropriate sexual conduct. And that's not very unexpected because the target area has both limbic projections and motor projections. So the electrical field will affect both of them. However, if we want to bypass this problem, you can envision, and this technology has been implemented, where you can control only the cell types of interest if you know them. So that's key. If you know them, you can control and access them and leave the bystanders unaffected. So illustrated here is optogenetic excitation, which uses a cation channel that's responsive, in this case channel rhodopsin, to blue light, and fluxes sodium ions in, which causes time lock millisecond precise action potential. So you can control it with very brief pulses of light. And you can elicit action. And the complementary tool is halorhodopsin here that pumps chloride ion into the cell and shuts off neuronal activity. Both of these tools have great value in removing the activity of certain circuits, or upping the activity of certain circuits, and we can use them to study the impact on behavior. And David showed you some of the great science that you can do with it. Interestingly, for these two tools, particularly the action spectra is well separated, so they absorb well separated wavelengths, which opens up the possibility of using both of them in different circuits or the same circuits for different actions. Now I want to go back at the beginning of the field and point to some of the challenges of tool development and the time it takes to incubate such tools to have maximum impact. The tools that I mentioned, channel rhodopsin and halorhodopsin, come from very simple organisms. So channel rhodopsin comes from the green alga here, Chlamydomonas reinhardti, and halorhodopsin comes from a bacteria, Neuromonas pharonis. They have somewhat fancy names, but people name them, so it's not their fault. And when we try to take these tools and move them into mammalian system, we are asking a lot from the proteins. We're asking that they are made in a proper way, in the right amount, that they're shipped to the proper location, and that they function. And without knowing and without understanding the system where the tools are coming from, we are impaired in our ability to use them maximally. So this is exactly the challenge that optogenetics had at the beginning. Channel rhodopsin worked well without major modifications, but when we tried to use halorhodopsin in the first in vivo experiments, particularly to understand the mechanism of deep brain stimulation, we encountered a very disturbing problem. The protein would aggregate and form these clusters that grow larger and larger and end up poisoning the cell and killing the neuron. So that was a somewhat effective but cumbersome method to damage brain structures, which was not the intention. So there was a lot of motivation because the currents were there. The thinking was that the protein is folding and acting properly. So there was a motivation to make the system better, but we had to understand what's the barrier. Why, when we put this protein in a mammalian cell, it doesn't work? So it ended up that knowledge about the base organism, the source of the tool, was crucial, because Neuromonas pharonis comes from a bacterium, and it doesn't have an endoplasmic reticulum, while mammalian cells have an endoplasmic reticulum. So we did this very simple thing. For a KDEL, which is a marker for resident endoplasmic reticulum proteins, in red here, and we saw that there was a 100% overlap of the clusters with markers of the endoplasmic reticulum. So the story emerged that this protein doesn't know how to get out of the endoplasmic reticulum. There's no reason for it to in its host organism. And then it ruptures. As we produce more, it ruptures, and then it poisons the cell. Knowing the cause allowed us to devise a very simple method to bypass the problem. And the method was, let's trick the mammalian cell into believing that this foreign protein, it's its own. So we took trafficking parts, very short sequences from mammalian proteins, and we attached them through basic cloning techniques. I shouldn't call it genetics, as we've heard. And we attached it to the sequence of the protein. So with many iterations, the end result was a protein that had an endoplasmic reticulum export sequence and also had a trafficking signal from an isoform of the potassium inward rectifier channel, which enhanced membrane expression. I didn't mention, to be maximally functional, these opsins have to be in the neuronal membrane. That's where they act to actually inhibit or excite neurons. So what you see here is a cross-section through a neuron that expresses an optimized form of the halorhodopsin, and you can see the very good membrane localization and lack of toxicity. So this development was possible keeping in mind the origin of the tool. So this results in safe, long-lasting inhibition, and also because the currents are larger, you can use the tails of the spectrum. So you don't have to use the peak wavelength, you can use the tail. And there's value in that because red light penetrates much better than blue light. So this strategy, very fortunately, could help other options too. So as they were developed, ARCH and MAC suffered from similar problems that could be bypassed by just taking this educated approach. So now how can we use optogenetics to understand more about the neuronal system? And there's a few ways in this, in which this might be possible. Is to perform experiments where we control the circuits. And we have all these options available with different wavelengths and different properties. However, we stayed behind somewhat in our ability to develop good controls. The current controls for optogenetics are fluorescent proteins, cytosolic fluorescent proteins. I opened the talk by pointing the need to study the aging brains. Aging cells are already weakened. Their membrane properties might not be as good. Opsins insert themselves into the membrane and they take up precious space from the endogenous proteins. And this complicates the situation if you don't have a good control that's actually sitting in the membrane, causes the same amount of displacement as the acting opsin. So that's why in my lab we were focused on this very, in a way I would call it small but big problem, to get appropriate optogenetic controls, especially for aging studies. So what we did was to use the information from the crystal structure that was available for channel rhodopsin, and also the knowledge that it's key that the opsin can bind retinal in order to function. So we mutated those residues, and we tested many constructs that would have good membrane expression, similar to the base opsins, but no photocurrents. So the result here for channel rhodopsin and ARCH is control opsins that don't have any photocurrents, although they have good membrane distribution. So we call them COPs for control opsins, or dead COPs for no current opsins. So we also express this in neurons, and they have very good expression. So just an encouragement to try to adopt controls for optogenetics. And now in terms of circuits, one way how optogenetics can help with diseases is to create models of the disease. I will put the caveat up front. To create an optogenetic model of the disease, you need to understand how the disease works. And there's very few diseases in which we know that. However, for Parkinson, we know the degeneration of the dopaminergic cells in the substantia nigra leads to the motor symptoms that we see. So what we can do is to deliver inhibitory opsins to the SNC, which is the area where dopaminergic cells reside, and to use combinatorial light to control different parts of the SNC and see, are all the dopaminergic cells within the SNC equally impactful on behavior? So let me show you some data around this project. We use stereotactic injection to deliver the inhibitory opsin, and then we confirm by staining that we are only targeting the dopaminergic cells in a Cre line. And we can use electrophysiology to confirm that we inhibit the neuronal activity in specific clusters of dopaminergic cells. And this is all very preliminary, but I'll show you a short video that illustrates that we can replicate some of the features of Parkinson's, such as tremor or freezing behavior. So there's no light now, and the rodent is moving around in its base cage. Inhibition of a cluster of dopaminergic cells in the SNC, the area that degenerates in Parkinson. And you can see the limb tremor on one side only because the stimulation is unilateral. So with light on, you see a freezing behavior from the rodent. How can this be useful beyond the animal model? So there's a few features for the optogenetic approach that can be very compatible with drug screening, with better drug screening. It's amenable to a variety of organisms. The delivery is viral. It's tunable. We try different light intensities, and depending on the light intensity, you can get different severity of the symptoms. It's fully reversible, so you can use fewer subjects in your experiment. And this opens up the possibility for educated drug screening because you know the circuit that you are acting on, so you can design better libraries. that's one way to contribute to neuroscience understanding, but that's only when you know the circuits. In many disorders, we don't. However, optogenetics can help us understand how some of the existing therapies work. So this is the case for deep brain stimulation. So we had a small project around understanding how deep brain stimulation works by using optogenetics. And this is highly relevant because in addition to Parkinson's, deep brain stimulation is also approved for tremor and dystonia, and more than 100,000 patients are actually walking with this implant today, and it helps their life a lot. And I thought it would be useful to show a patient, because we talked about the relevance of patients contributing to the knowledge. So I'll play a video. Hopefully it will connect now. Good.
Desliga o DBS. Você quer ver o que é o sofrimento? Tem essa coisa, olha só aqui, ó. 2 segundos.
So that's a battery pack, and he's deactivating. Nice.
Fica tranquilo. Agora eu estou com Parkinson. Real, é verdade, não estou mentindo. Isso aqui não é o Michael Fox com o limbal aqui.
Liga.
Isso aqui é Parkinson. Agora vou ligar de novo.
Reactivate the stimulator.
Acredite se quiser.
So it works very well. However, there's side effects for the reasons we mentioned. And although there's a significant amount of research, we still don't understand to date how it works. And it would be highly relevant that we understand so we can adopt it for other diseases as well. And it's been approved for obsessive compulsive disorder, and there is basic research showing that it could help with depression as well. So the study that we did around this was to use optogenetics in the centers that are stimulated and try to mimic the results, but in a cell type specific fashion to understand which circuit elements are responsible for the disorder and the therapy thereof. So by injecting a virus only into the subthalamic nucleus, which is the target area for DBS, you can get, in a way, genetic extraction of that area because the nucleus is embedded in an inhibitory network. So if you use an excitatory promoter, you only get expression in the subthalamic nucleus, and you can control that area. Or you can avoid the cell bodies altogether and only get expression in the axons. So this is always very powerful because it allowed us to compare the effects on stimulating the cell bodies versus stimulating the axons. And what we've noticed is the cell body stimulation was not effective to recapitulate the effects of the therapy, but axonal stimulation was. And also at the electrophysiological level, we saw a duality. When we used high-frequency stimulation, we abolished activity in the nucleus, while when we use low-frequency stimulation, we introduce noise into the system. And this is an interesting fact, because in clinics, when the neurosurgeon implants the electrode into the subthalamic nucleus of the patient, high-frequency stimulation gets rid of the tremor, but low-frequency stimulation exacerbates the tremor. So it was very interesting to see this duality in electrophysiology in our rodent data. And it pointed us to the fact that we need to understand where these axons are coming from. So we identified the source, the likely source of the axon superficially onto the cortex, and then we performed the stimulation onto the cortex to see, can we mimic the effects of deep brain stimulation by stimulating superficially into the cortex? And this is only one example of a light path from a rodent, a Parkinsonian model, without optogenetic modulation, and here with optogenetic modulation of the cortex, so not deep brain nucleus, but of the cortex. So now the question is, how relevant is this for the human brain? You start with a very small mouse brain, and this is a circuit that's not fully described. So the neurosurgeon that I was working with at the time, intrigued by this so-called hyperdirect pathway, and pointed by the need that he had to refuse many of the patients because of the hemorrhage likelihood. So many patients, because of the deep brain target, they were likely to get hemorrhage. So he couldn't include them in the trials. And so he was motivated to see, is this pathway functional as well and present in humans? So this is a DTI scan that shows the white matter tracts and is done on a Parkinsonian patient. And it points to this pathway that we suggested in the rodent brain that comes from the cortex and concentrates onto the STN. And by using subdural microelectrode arrays, Jamie Henderson was able to confirm that this is a functional pathway. When the electrical stimulation was placed in the STN, it could actually cause activity onto the cortex. So this opens some interesting possibilities for TMS or electrical stimulation of the cortex, and it's one way how basic research experiments could educate some of the developments. But it also points to an insufficiency of knowledge about axonal tracts in the mouse or in the rat or in the humans. We have very good maps now thanks to the Allen Brain Institute, and we also need functional maps, which will happen soon. And in terms of the anatomical maps themselves, we do need good maps that deal with axons. And a technology that recently came out of the Deisseroth lab at Stanford, Clarity, could be helpful in generating these maps for the axons. If you base your axonal mapping or slicing technique, the risk of misaligning the tracks is very high, so we need an alternative method such as scale or Clarity to be able to track such axons. So briefly, since it's published, I won't mention how the technique is performed, and I will take maybe 1 more minute. There are, however, some caveats with Clarity as it is now. Registration is one of them due to swelling and shrinking, and antibody penetration is another one, and also browning due to the Maillard reaction is the same reaction that made your pastries brown in the morning. So reversal of these would be useful, so our lab is further working on developing Clarity. Meanwhile, though, passive diffusion on thick slices works very well and can allow you to antibody stain 1 millimeter thick coronal sections and also image them. So I'll show you one example of here. Here are coronal slices cleared to passive diffusion, and it takes about a week to clear 1 millimeter thick slice. And then you can embed this in antibodies and allow for passive diffusion and stain it. So we stained for areas that we are interested in terms of neurodegeneration. And you can notice, for example, here in the SNC, you can notice the axonal tracks going to the striatum, so you can map them without the risk of misaligning. And what's interesting here in a PPN, another area that's degeneration prone, you can see very sparse dopaminergic population, which you might lose if you look only at thin sections. So here is how Clarity could help in terms of mapping the axons with further development. We could start with 1 millimeter thick slices, make the method better, so we can get better maps. In the interest of time, I think I'll, let me go to the acknowledgement slide. And I'm happy to talk more about Clarity development with interested people, but I want to acknowledge my lab at Caltech, and I talked today about the protein engineering work of Claire Bedbrook, graduate student in my lab, and Bin Yang working on the Parkinson project and the Clarity development. And I'm very grateful for research support, without which our work would not be possible. So thank you so much for your time. It's time for questions and for work.
I thought your work is very interesting. I was very intrigued by your DTI projection that you showed that the M1 has a projection to the sub-TN. I wonder if you could illustrate a little bit more how, when you put the opsin into a subthalamic nucleus, what is the input from motor cortex in terms of the inhibitory and excitatory network?
Exactly. So what we noticed is that layer 5 pyramidal cells project into the cells on the subthalamic nucleus, and those projections are excitatory mainly. However, there are also local inhibitory axons that could be stimulated by the electrode itself. So we used Cree lines for GAD and parvalbumin to try to target some of those inhibitory elements in the area as well, but their modulation was not as effective as the hyperdirect pathway to the subthalamic nucleus. So it's a rather complex picture that would require a longer discussion. But there's many cell types. It's not, I presented one angle. There's many other angles in there that I didn't have the chance to talk to.
Recall correctly, your original experiment where you unsuccessfully tried to replicate the effect of HFS on STN by silencing with Halo, right? That was using early generation versions of Halo and lentiviral vectors. Have you gone back now with your improved halos and with AAVs where you get high levels of expression and efficient silencing and repeated those experiments to see whether you see any effect at all?
Yes, actually that work was done in the Deisseroth lab, and I was not involved for impartial reasons, and it's not as effective as cortical stimulation. So even with the 3.0 generation, the improved protein that I talked about, and complete inhibition of the STN, we could not get the level of effect that was due to the axonal stimulation.
But some effect could be seen.
Very modest, very, very modest.
And so what's your explanation now for your current explanation for why in humans the DBS in the STN works? Is there no effect to inhibit the local STN neurons? Is it inhibition of the descending cortical projections?
So that's why I said that we don't yet understand the mechanism, because the picture might be more complex than that. Since we published the study, there was actually a follow-up, beautiful electrophysiological study that did this. Didn't use any optogenetics, but stimulated with electricity in STN and record single-cell activity from the cortex. And what they noticed is that low-frequency stimulation was synchronizing those cortical cells due to the backpropagation of the action potential from the STN. While high-frequency stimulation, interestingly, there was more failure of the backpropagating action potential at high frequency, so those cells would not be synchronized. This ties in with the human data where losing dopaminergic population creates beta waves in the cortex, 20 hertz, and dopaminergic medication could restore that. So the emerging picture is much more complex, and it would require more work to understand, but it might not be a simple issue of we inhibit this or we excite this. It might be an issue of communicating brain structures and how do you communicate meaningful information. Having areas synced up and firing at 20 hertz is not as useful for the animal as having a complex code. So 20 hertz might enhance that perturbance of activity versus high frequency. So it's a complicated picture, and for this we need the maps, we need ability to record from single cell that we genetically identify as well. And through that we might get to an understanding.
I was wondering about your Parkinson model. So there you saw unilateral tremors in the mouse when you stimulated the substantia nigra neurons,
Yes.
right? Have you done, or are you planning to actually add an additional component, namely, for example, the alpha-synuclein mutations, where you actually would have a pathology evoked by a causative gene as well? Because one can question whether you really have a Parkinson model.
Yes, that's a very good point. So Parkinson is a evolving chronic disease, and as you lose cells, there's plasticity changes in the brain. The model that I showed is missing all of those aspects. However, is adding the understanding of if we remove this dopaminergic population, how does the brain change versus if we remove this dopaminergic population in the same nucleus. So I would say that there's two different sides, and one approach is good for one and not for the other. In terms of the chronic effects and the plasticity changes, a genetic model might be more appropriate for this.
But you could cross in a genetic model quite easily into your animals, right?
Yes.
Well, one of the most fascinating aspects about the circuit that has always puzzled me is that, and I've witnessed this in the operating room, so you can put the electrode in into the STN, and immediately the tremor ceases. I mean, within a second or 2 seconds. You leave it on for 3 years, and it stops, and you turn it off, and it immediately comes back. Now, we know that the brain is plastic and synapses adapt, but I don't understand. I mean, is there any inkling why this should be so, why there's a complete lack of a compensatory adaptive response to this regimen of stimulation?
So that's a fascinating phenomenon, because if you do this at 3 years, it might come back. But if you turn it off at 5 years, it might not. So Jamie was telling me that there are cases of patients that come back for an electrode tune-up, but they don't notice right away. So if you had the stimulator for long enough, and maybe 3 years, I don't know where the threshold is, but if you have the stimulator on for long enough, it could malfunction at some point and you wouldn't notice for days maybe. So there are, I guess one answer is that there are changes, but we don't know how long they take and what kind of changes they are. But there are plasticity changes. And then some very preliminary reports suggest that in DBS-treated brains, there's more surviving dopaminergic cells versus L-DOPA-treated brains, which raises the possibility that electrical modulation of the system could have some beneficial effects onto the cells themselves, maybe by reducing excitotoxicity or by releasing growth factors, which is research that we have from auditory system with electrical stimulation. Neuroprotection was documented in that system. It's not being done as much for deep brain stimulation in Parkinson due to the barriers. There's human, yeah.
During your intro, you mentioned that you were going very specific, and of course you did. I'm just wondering if you can just zoom out a little bit and just sort of give us a more global picture, for example, with Parkinson's, and whether or not in the future we're looking at an intervention which is not a variance on deep brain stimulation to another intervention which is using light, photonics or something of that sort.
So I like to stay close to the data, but I'll
yeah, no, we're going to speculate.
I'll try to take on your call and do the jump. So one way how optogenetics can help is to make us understand the circuit so then you can design better drugs. So that's one way. If you are pushing me even further and thinking about gene therapy and fiber optic wouldn't be so big of a concern. You saw how invasive the electrode itself is, so that wouldn't be a big leap. The delivery of the genes, though, that's the one that has to be approved and proved safe, and we have to do this in larger animal models to prove its efficacy and safety in animals very close to humans before we make that leap. So, but it's not impossible.
All right. Well, let's thank Viviana again.